You have a camera too then.
Yes. I don't know if I need my laptop, but if I, you know.
Erica, can you hear us?
Yes, yes, I can hear you.
Okay, my sound is off. Can you say something again?
I can hear you. Can
Yes,
you hear
I
me?
can hear you too.
Yeah, great.
Good morning, Han. Or good evening in your case.
Hello, hello. Yes, it is evening.
Is it horrifically hot there at home?
Sorry?
Is it horrifically hot? I saw that we had like a heat wave warning.
I turned on the aircon, so I'm currently okay. It was very.
We don't have that issue over here. We had snow last week. Hang, according to the to the agenda, University of Sydney is chairing. So would you be willing to chair?
Yeah, that's fine.
And University College London, Matt is taking minutes. Is that okay?
I'll probably catch up with the recording.
Okay, yeah.
But no problem.
Okay, then I leave it up to you. So Hang, you can go ahead.
All right. Are we expecting more people to join? I think there's only, is that all of us? Doesn't looks a bit more quiet than usual.
Yeah, we are all together in one room, so that saves also a few faces, I guess.
And Andrea said she couldn't make it specifically, so we won't see her.
Just to check, is someone able to switch on the recording?
I am recording.
Great, perfect, thank you.
Yep. All right, looks like we've got quite a bit to go through, so let's get started. So I'm gonna be just, let me have my agenda on. So all right, so just starting with housekeeping. So everything that is remaining on the master list is to be kept, and no meeting next month. So that's right. That will be holidays. Series 1. Matt is finishing up some parts in the paper. I think I see in the comments that there's also things for, I believe, me and Dimitri to finish up as well. So shall we go through that first and then go to series 2? Is that okay with everyone?
Yeah, I mean, I've tried to make it to sort of divvy up the last little remaining things to people, so everyone's clear what they've got to do, and hopefully it's straightforward. The draft is fine, apart from those little things that I mentioned. And once those are taken care of, then I think it's good to go.
Yeah.
But no, I mean, you run through them one by one, unless there's something there which is confusing or someone wants to talk about.
Okay. Oh yeah, I'll just quickly go through them one by one. So in terms of references, okay, there are some problems. Okay, I think this one is for Wendy.
Yeah, I was putting in the chat or in the thing. I will do that because it's simply something I need to reformat in EndNote. So then we don't have to do this all manually.
Okay.
I guess we might want to do it right at the end because there's a chance that there may be a couple more references if we find other papers that we have to reference for the experimental section. So I guess that's the last thing we should do.
Yeah, so when all other things are finished, please let me know. I will then update the references in EndNote and make sure that the DOIs are correct, because I also noted with other papers where we needed to add the DOIs that the computer not always does that correctly automatically.
Yeah,
So I have to correct
also
them in
happens.
EndNote.
That also happens when I did my references for my thesis as well. It doesn't automatically load all of the DOI, especially older papers. Is a bit hard as well. Finding
Yeah,
them.
and sometimes there's a duplication in the DOI, which makes it look very weird. But usually in one single paper, it's not that many references you have to reformat, and the ones you mostly see, they will be have been reformatted by now.
Yeah. All right, so we'll just leave that for when we need to do towards the end. Diagrams. Okay. Any remaining chem drawers? I'll have a look at the chem drawers, Matt. I think I put all of them in. I'm not sure if anything's. Is there anything particular missing that you want me to go look for?
No, not at all. It's just I wanted to just check we've got the whole set. I mean, I know you've already put a lot there. I just want to make sure we've got the whole lot.
Okay. In terms of consistency, I'll go and have a look at some of these. These are I think mostly typos. So I'll have a look and I'll try to resolve them as much as I can. Okay, supporting information from this spreadsheet. Supplemental files. Oh, the supplemental files. I think that is me. Page 55. That's supposed to come from me. I'll add that one. That's me. My spectra are all in the SI NMR sample, 25th July. I think Dimitri's ones are also in there already. So that's all that should be all for this paper.
Great, that's fantastic. If all the NMRs are in there in that file and it's ready to go, then that's awesome.
Yes, I have also got my offline lab archives on the repository for UCIT. So I've already got a link there. So that's that's for me. I'm not sure about Dimitri.
Is that in the paper, that link?
I don't think I put in there yet. I'm just gonna put it in as well.
Okay, yeah. I mean, if you wanted to dump that link into the just into the GitHub repository there, then I can copy and put that in the thing. That's great.
Okay, yeah. More typos from me. Okay, and literature references for compounds. I'll go into that one.
Yeah, so that I just checked one simple one and found that there was an NMR there. So I just wanted to make sure that we have found them all.
Okay.
Yeah, that's it.
Yeah. Acknowledgement. This one I'm not sure for use it. I'll probably have to go and ask Peter and Alice, but they've also said that when you are done, I think there's one more stage where you just give the draft out to everybody. So I think that's when Peter and Alice are going to look at this and potentially add this in. I haven't confirmed this yet, but they haven't touched this draft is because there's still that supervisors, everybody's review stage. So I think they've been waiting for that. Is that okay if potentially they can add that acknowledgement or look at it when that comes around, or do you want that for?
Well, I mean, it's just the acknowledgements bit, right? So, I mean, if there was anyone at UCIT who's contributed or should be thanked, or if there's any funder that should be acknowledged, it's really just that.
Okay.
So, yeah, I mean, if you could, I mean, I think the draft, you can give them the draft now and just ensure that the acknowledgements is complete.
Yeah. Okay, now I'll give that to them next when I'm gonna meet Alice tomorrow, so I'm gonna let her know. Okay. All right, so there's the miscellaneous stuff, capitalization.
Yeah, if I look at the miscellaneous hung, the last
Yeah.
comment is, what is our conclusion regarding log D? It was true that in vitro, in general, compounds with a log D higher than 2.5 work better, but in vivo lower than 2.5. I think that this is simply due to the way the fungus is appearing. So in vitro, you have loose hyphae, so it doesn't need to penetrate much more different barriers. So the barrier is only the fungal cell wall and the cell membrane. In vivo, you have the mycetoma grain. So probably the compounds need to possess different properties to be able to penetrate through the host cells, then the cement material, and then it needs to penetrate the fungal cell wall and fungal cell membrane. So probably the difference there is the penetration throughout the cement material. And that probably needs different properties than only crossing the cell wall. And I think that's the reason behind this discrepancy.
Right. Yeah. Yes, exactly. So I just wondered, would you be happy to convert what you just said into one or two sentences in that bit of the paper?
Yeah.
I think it's really important that there's a balance there, and it's a pretty fine balance, I think.
Yeah, I will make a note. I will edit it and put it on me.
Great. I mean, in the bit, I think in the bit of the paper where, I mean, either in the conclusions maybe, or in the bit where you talk about the in vivo testing and the correlation with log D, it would be, it'd be great just to explicitly state that, because I think it's it puts quite a constraint on the design of the molecules. Actually, it's quite interesting how narrow that window seems to be.
Yeah, and it's also opposite. So that means that you always need in vivo or a grain model to screen, because you would not select compounds the same way in vitro as you would do in vivo.
Yeah, exactly.
All right, so it looks like that is all for the manuscript. Now, going back up to series 1, I will add that part about the relevant part to the wiki of the virus series. I'm doing places correction right now, so that's why I haven't put that on yet. But when I'm done with it, I'll divvy it up and put it into the right place. So just leave that task on me for now. Series 2 aminothiazoles, there are some comments on here, but is there any particular action or any question for this particular section? So there's things like compound 771, 772.
Okay.
Yeah, hi. It's Echo.
So just to quickly talk about that, I think it's probably not that useful to talk about the Uni of York hits at the moment, because nobody from the Uni of York is here. But basically, they sent us a fair few compounds a couple of months ago, and the IC50s for those are nice, but there's only 2 that worked. And then I've just updated the SAR page for all the aminothiazoles to try and capture everything that's in there. It's not messy, but it's hard to organize, because we now have quite a few compounds that are a bit like, orphan is a bit strong, but there's not necessarily clear themes, I guess, to organize them around, but at least they're all in one place. So that includes the one that Bernhard had sent that we didn't have on there before, and then some of the ones that the Uni of York is planning to make with amide and double amine linkers. And then, last time I talked about contacting this group leader, Marco Pieroni, and we did contact him, and he sounded keen. But then once I, responded back to his response, I haven't heard back for 2 weeks or so. So initially that was positive, but I don't know where it's gone from there.
Maybe simply send a reminder.
Yeah, I'll check how long it's been since I sent the response, and then maybe, yeah, send a reminder before the end of the year. But yeah, that's basically everything going on with immunothiazoles, at least on our end.
Okay, that sounds good. Okay, so
And maybe,
Yeah.
from our end, we did just a bit of search in the PDB database using the aminothiazole framework, so the small aminothiazole, framework, and also the bigger framework of our molecules that have activity. I think is Reza on the call. I believe, I'm not sure. So this is a student from the UCL MSc who did the search and uploaded the Excel file on GitHub just to see which targets we can see aminothiazole compounds interact with in terms of crystal structures, but also in terms of predictions. Not much. So if you look at the the Excel file, there aren't many hits, but it may be because there are no co-crystal structures of aminothiazoles with targets at the moment. Let me see if I have it open. So the biggest hit of our molecule was the Mycobacterium tuberculosis transcriptional regulator ETHR. Wendy, does that sound familiar? Is it something we can test against?
Yeah, a transcription factor is always a little bit difficult, of course, because it influences many things. So that's maybe also the reason why you have different hits to begin with. But Mycobacterium tuberculosis is a bacteria and a fungus, so a little bit further away, but it's more closely related to the actinomycetes, of course. But yeah, it could be something to check. Yeah, I had experience in the past with expressing proteins of Madurella mycetomatis. But yeah, this is a transcription factor, so I'm not sure if that would work.
And we do know that immunothiazoles work against tuberculosis.
Yeah, we do know that. Yeah. It was just we were hoping we may find some hits that were maybe new that we could test, but there aren't many hits from the PDB database. So maybe our best way forward to try to elucidate some mechanism of action of these molecules would be from maybe literature, like reading if there are not many available structures.
Yeah, what I recall from the literature is that the aminothiazoles have been reported to have many different targets.
Yeah.
So that makes it also a little bit more difficult.
Difficult, yeah. I've read the same, yeah. Yeah.
But maybe then indeed it is a transcription factor in the end, because then you influence many different targets. But yeah, I don't know how to test this actually.
Yeah.
It would have been easy if it was a simple enzyme where it reacts with, because that's not that difficult to do. But
yeah, no, that's okay. Yeah, we can focus on the literature and see if we can find a target that is easy to do. I mean, the HDACs are easy to test. Just need to look more into the literature and see if we can find a more strong link. With
and maybe it's also something we could do for maybe one of the other series to see if there's a more clear-cut correlation with an enzyme with one of the other series.
Yeah, you mean the search of the PDB database?
Yeah, like for instance, if we do this for series 3 or series 5, then maybe we can take this angle to another series.
Yeah, I think it's a good idea. Yeah. And with GIF, you've seen I sent that molecule to Desiree for their next target. Clementine, I haven't assigned a MycetoS code. I'm not sure how. Yeah,
Normally, we haven't assigned a MyOS code until they've
maybe.
at least started making it, and ideally
Okay,
have
so
made
that's
it.
fine at the moment. Okay.
Yeah.
Sounds
Just so
good.
that we don't end up with lots of codes that correspond
Are
to
not active. Yeah. Okay. Yeah, and these are the updates from our side.
All right. I guess that's everything for series 2 for now. So I guess we'll just move on to series 3. So one question for in vivo testing for myOS 6.2.6, 6.2.8. Do I want to talk about this? Because it's also involved University of York, which they're not here.
Yeah, so we simply don't have enough to test in vivo. So if these results are still needed or of interest, we need the new compound. The larvae season ended, so that would, if we get these, that would be for next season.
Yeah. Okay, and for series 5, Andrea, Ma, and Charlotte have been working on a draft for the benzoxazole manuscript, and Clementine has searched for the MMV box for benzoxazole. So these are also just information. Is there anything that you want to talk about this, or this is more like FYI?
It's a little bit FYI because it's ongoing, and yeah, we will report back when when it's needed.
Okay.
So yeah, it's in the drafting phase, in the manuscript phase.
Okay. And is that also the same for series 6 as well, Matt? Dimitri is still working on the other manuscript as well. How is that going?
Yep, that is happening. We will be able to spend more time on that once we've got the Phenaromel manuscript in.
Okay. Looks like there's many things going, especially 3 manuscript for mycetoma. Sounds good, but so is there any other business? Oh, that's a very short list today. I just realized. Or that way I'm just rushing through this.
Yeah, for the other business, we now are at the end of 2024, and next meeting is cancelled because that's on Christmas Eve, I guess.
Yeah.
I will make the new schedule. I assume it's still University of Sydney, University College London, and us in the rotating of the convening, making minutes. Or do we need to include others, or is one of the other centers not able to do it anymore? Because that will help when I prepare the roster.
Uni of York, maybe?
It would be very nice if we could involve if people for like Andrea and people from York are interested in contributing. That would be really good. We could ask them perhaps if they're willing to do that.
Yeah, because last time I asked Bernard to participate, because I think it makes it more of a project of all of us and not, it's not that much work. So if anyone would like to participate, please, shall I make a note on GitHub to see if they are interested?
Perhaps with people like
Yeah.
Andrea could say, like, because I assume she won't want to do it when it's the 11 p.m. meeting
No.
for her. But if you can assure her that you can organize it so it won't be that way around.
Yeah,
Thanks for doing the schedule, though. That would be really good. And I guess, we should try and keep with these 2 times for the meetings just to make sure that everyone's got some availability.
Yes, and if the university, if some universities of the United States or whatsoever, if somebody would like to participate there too, then they could do the easy times for them.
Just on any other business, Wendy, you and I were talking about whether or not there would be any purified protein samples from Madurella that we could put into this affinity selection mass spectrometry screen that I was mentioning. I don't know if you have any more thoughts about that, about whether there may be anyone who has a couple of milligrams of purified protein we could put into that kind of screen.
Well, I don't think anyone else did any protein work on Madurella mycetomatis except us. It has been very difficult to get proteins from Madurella at all because Tim here tried several times. Certainly not milligrams. The only thing what we can have in larger quantities are the ones which we recombinantly expressed. And those were, TCTP, so the translationally controlled tumor protein, fructose bisphosphate aldolase, pyruvate kinase, those we have. But yeah, these are recombinant proteins. So if they fold exactly the same as the original, I don't know.
Yeah, it was really the kind of understudied or not yet studied recombinantly expressed his-tagged proteins that they're interested in. So something which is new. And I guess my question really was whether there's anyone who happens to be looking at one of those new targets for I don't know, for other reasons, or maybe is interested in getting a screen done. That was really my question.
No, yeah. So we do have those because we discovered those as potential antigens. They were antigenic. So I still have the clones. So it's not that easy or not that difficult to express them and purify them. So, but that's only those three. We do have the protocols to generate others. But then we need to look at which proteins we would be interested in doing, because it was quite easy to do this actually. But the recombinant stuff, to isolate native proteins, that was a nightmare.
Okay. I mean, I just wonder whether we should approach DNDi or something to ask if they could, support, because you can buy these proteins, right? I mean, you can get them made in industry, right? And if we decided there were 3 targets or something that were interesting that had never been made before, we could see if we could generate them. Because if we do generate them, we'll be able to get the screen done. Okay, let's talk offline in that case and see if we can do that. I guess I'm just conscious with the discussion about the aminothiazoles. I'm just conscious that it'd be really nice to be able to try and target something which has not been targeted before.
Yeah, maybe the Warnin body, that's still an interesting target, I guess.
Exactly, exactly.
Yeah, and we do have the proteomic and transcriptomic data, so we could easily select a few targets. Also, the trialose pathway, for instance, is present in fungi, but not in human.
Yep, yeah, exactly. You see, as soon as I mention it, you see you've got targets already.
Yes, I do know the targets, but I didn't express them yet because we had so many other things to do.
All right, is that everything? I do have a question, Matt. In terms of the timeline for just finalizing the draft for Series 1, when do you kind of want that back? Especially for my thing, because what I'm doing is I do have a deadline on my thesis correction, which I really need to get that done ASAP, to get that finished. I think I might need to do that, prioritize that before checking to this draft. But there are also some really quick thing, which I can do for the manuscript right away. The SciFinder thing might take a bit of time, so I won't be able to finish that, but other stuff sounds a bit quicker. When do you want this back to you?
Yeah, I mean, as soon as you're done, we can submit it. I guess, yeah, there aren't the things in there aren't big, and the SciFinder thing, I guess I assumed you would need that for your thesis anyway, right? Because if there's any of your compounds in there, then you're going to have to highlight if they've been made before. So yeah, I mean, as soon as you're done, we are good to go.
Okay. Yeah, I'll get that back to you as soon as possible. I just need to actually do the correction for my thesis first and then get to this. That's all. All right, so if that's all, then we actually have a very short meeting today. If there's nothing else, I hope everyone have a nice Christmas/holiday season for next month. And we'll see you back all next year.
Merry Christmas.
Merry Christmas.
Merry Christmas. Happy
Merry
holidays.
Christmas.